All right, at the end of last class, we got to one slide where I needed to make a few more points. So what is this that I've drawn so far? Replication bubble, okay. And in red, which enzyme creates the primers that are used to initiate DNA synthesis? Enzymes usually end in what? 3 letters? ACE. And these are primers. So yeah, this is the enzyme primase. And if we label these with polarity, then we can zoom in and we can figure out the polarity of those primers. Because they're going to base pair in an anti-parallel fashion, as you all know. And then you get synthesis of new molecules of DNA, new strands growing from the 3' end of the primers. So the primers are in red, DNA is in black. So I want to show you what this looks like a little bit later in the process. While this chromosome is being replicated, that replication bubble expands. But let's say the replication hasn't completely reached the telomeres yet. So there's still a little bit of double-stranded, unreplicated chromosome there at the telomeres. Those original primers are still there. And we have, again, this is review, leading strand synthesis. Synthesis started at those primers at the original replication bubble and just cruises and cruises and cruises on the template strand of DNA. Get a new strand, again in black, it's just longer. Right, so here they were short, the replication bubble was small. Here that same molecule of DNA polymerase that's copying the DNA is still chugging along the chromosome, copying or creating, synthesizing the second strand based on the first strand as the template. And we talked about now that there's some single-stranded DNA, part of this replication bubble, primase can come in again and create more primers, and that allows more polymerase molecules to come in and start synthesis of new pieces of DNA. So the replication bubble grows and grows, primase keeps adding these red RNA primers to single-stranded DNA. And by the time you get to the end of the process, once replication has reached the telomeres, this is what this looks like. We've got the leading strand, started at the replication bubble and synthesized all the way to the ends of the telomeres. And then you have a number of, usually more than 3 or 4, RNA primers that have initiated synthesis. And those are the lagging strands or the Okazaki fragments. I should have capitalized Okazaki because that's named after the person that discovered these fragments. So that's where we got to the end of last class. Leading strands, lagging strands, Okazaki fragments. But we're not done yet. We need to tie up the loose ends. So what are the problems that exist with this molecule, these 2 molecules of DNA?
There's that little snippet on the bottom strand.
Yeah.
Far right.
Bottom right.
That's kind of just there.
In blue, circled in blue. What's wrong with that?
It's not being copied.
What's wrong with that? Why is this a complete DNA molecule? Is this the whole chromosome? Has it been replicated? What's happened? Why hasn't that part of that chromosome been copied?
Because there may not be a primer that matches that specific region, so it goes to the next closest one, which means you might lose those nucleotides.
No primer. There's no way for primase to create a primer that's floating off in the nucleus there to the right of the chromosome. So there's no 3' hydroxyl that's part of a primer molecule. that could be used to initiate synthesis that would start to synthesize into the end of the chromosome. That's why our telomeres shorten every time the cells divide, because there's no way for primase to create primers that let the ends of the chromosomes be copied. So if that's true, does this also happen to the other strand, the other template strand? Right, upper left, there's a little tiny piece there that hasn't been replicated. That's one problem. So our telomeres shorten every cell division because the process that copies our chromosomes actually can't copy the ends of the chromosomes. The lagging strand, note, ends of the chromosomes. Leading strand is fine. Get synthesis right to the end of the molecule. What are the other problems with these molecules at this point? Two more.
Are they already hydrogen bonded?
So these are base paired. Right, the new strands, the newly synthesized strands are all hydrogen bonded to the template strands. Wait, what are those newly synthesized molecules? Are they DNA? Is this double-stranded DNA now? What's the color coding up here? What's red? Primers are DNA or RNA? Primase makes RNA primers. Everything that's drawn up here in red is RNA. Have we synthesized new DNA chromosomes yet? No. There are primers, there's RNA base paired with our DNA. Here, and there, and there, and there, and there, and there, and there. Everywhere there's a primer, now we have DNA-RNA base pairing. We've got to get rid of the RNA so that we have two strands of DNA, a double helix that's just DNA. And depending on whether you're talking about a prokaryote or a eukaryote, there are different enzymes in our cells that go ahead, after replication finishes, they chew up the RNA and they replace it with DNA. So that's the second problem. We've got end replication problem. We've got the fact after synthesis just finishes, there's still some RNA left, has to be replaced by DNA. And then what's the third problem? On those strands that were newly synthesized, how many pieces of DNA are there? How many have we drawn here? There's a leading strand, that's one piece, but it's not covalently bonded to these Okazaki fragments. Right. There's gaps here, yes? Look, the arrow doesn't touch, it's not physically bonded to that primer. That primer gets chewed up, replaced by DNA. We have to actually physically covalently bond those two 5' and 3' ends of the same strand together. We have to link those together to make one entire strand that goes from one telomere to the other. Does anybody remember which enzyme does that? It's another one that ends in ase. What is it? I heard it, ligase. You're right. This is DNA ligase. Ligate, tie. So DNA ligase comes along and ligates or ties all of the short fragments of DNA together to produce one strand. It's got a covalent backbone that goes from one telomere all the way to the other. So those are three problems we have to worry about at the end of replication. So that's the end of replication. Now we move on to the new subject, meiosis. Here are the goals, basically. We're going to talk briefly about the top 2 points, and then we have a few class exercises to do today and probably next class as well. What's the point of meiosis, please? Why do some of our cells do it? Create haploid gametes. Okay, so did everybody in the back know that too? Meiosis. Start with if you're a diploid organism, you wind up with haploid gametes. Why is that?
So you can combine genetic information.
So you can combine genetic information so that we can have sex and reproduce. What happens, but why meiosis? Why don't we just do mitosis and create gametes from mitosis? They'd end up with double the chromosomes. So that's the why of meiosis. Why do we bother doing meiosis when I could make things easy on your lives and just say all you have to know about is mitosis? So if you start out with a diploid cell, it's got 2 copies of every chromosome. You go through synthesis, you have a cell with 4 copies of every chromosome. If cell division in meiosis only happened once, how many copies of each chromosome would be in each of these resulting daughter cells? 2. 4 cells, split the cell in half, you get 2 chromosomes in one daughter cell, 2 chromosomes in the other. So let's say, instead of the way we know meiosis works, where these cells would divide again to produce, so in red, each of those cells normally would divide again in meiosis, that's the second division. And you'd get 4 cells that each have 1 copy of the chromosome, 4 haploid gametes. So why is it so bad? Why can't we just stop at the 2-cell stage, do mitosis, and there's a sperm, and here's an oocyte? What happens when we combine the sperm and the oocyte together if you don't do meiosis? How many copies of every chromosome does that 1-cell embryo have? If you've added 2 and 2 together, you get the F1 generation, the offspring, then would have 4 copies of every chromosome instead of 2. We have to do meiosis to divide cells in half and half again so that when those 2 zygotes, when those 2 gametes come together, a sperm, say, and an oocyte, then you regain diploidy. 1 chromosome from the sperm, 1 chromosome from the egg, you get a diploid again. So presumably that's why we do meiosis. There is no why to it, of course. It just happens. It's not like our gametes have intentions. They know what they're doing. But that's what happens. That's the benefit of meiosis. Synthesis, that's exactly the same as mitosis. Start with a diploid cell. It doubles its DNA content. Mitosis, 1 division. Meiosis, 2 divisions. Any questions about these basic concepts about meiosis? Then it's time to get together with your close and good friends that are sitting next to you and share your answers to this question that was posed at the end of the video for today. So if you have a cell that's got heterozygous for chromosome A allele 1 and chromosome A allele 2, and you've got 2 copies of chromosome B that have allele 1, what happens after meiosis? So take 2 minutes, share answers, or figure it out if you haven't worked on this yet. And let's talk about what would you draw in the 4 gametes that are produced after the 2 divisions of meiosis. So get at it. We'll talk in 2 minutes. Punnett squares? No, not Punnett squares yet. What have I not drawn up here that would be useful for answering this question? So sequences could be useful for figuring this out. Just allele nomenclature. So what hasn't happened yet in this cell on the left, upper left? Right, haven't drawn it as having gone through replication. So what does this look like after replication? You have 2 copies of chromosome A allele 1 and 2 copies of A2. And you have then 2 copies of B1 from one parent and 2 copies of allele B1 from the other parent. So that's post-synthesis. You've got 4 copies of every chromosome. There's 4 chromosome A's and there's 4 chromosome B's. Now, this is the really important point about meiosis. What happens during the first division? So the cell's going to divide.
The sister chromatid separates.
Something separates. That's the critical question, especially for this part where they're heterozygous. Got one copy of allele 1 and one copy of allele 2. How does that first division break up those two? The 2A1s go together, and the 2A2s go together. And because we're talking about a homozygote for chromosome B, it doesn't matter. Half of the, well, it does matter, but you don't have to make a decision about how to combine the A chromosomes with the B chromosomes. The only thing you have are B1s, allele 1s of chromosome B. Okay. Then after the second division, not the sister chromatids, but the chromosomes, the A's segregate equally. So you get an A1 in each myocyte and a B1 in each. That's division of that top cell. And division of the bottom cell gives you A2s with B1s. You could, I don't know, is it useful to use a Punnett square to do this? This is setting up a Punnett square. This is not filling out a Punnett square. And we'll talk about that next class when we start talking about what Punnett squares represent and how Punnett squares work. So don't worry about Punnett squares yet. Meiosis is the introduction to the use of Punnett squares. So those are the 4 haplotypes, the genotypes of haploid gametes that result from that meiosis. Asking about ratios of these 4 gametes that are circled in red on the right side there. What's the ratio of the haplotypes of these gametes? If I made that an exam question. How many different haplotypes are there? There are 4 cells, but there are 2, what are the 2 haplotypes? A1 semicolon, because these are different chromosomes. Semicolon means we're talking about genotypes on 2 separate chromosomes. There's an A1, B1, those are there and there. And then there's A2 semicolon B1. Those are the bottom 2. So there are 2 different haplotypes. This you could call a 1-to-1 ratio. There's an equal number of gametes with 2 haplotypes. For every A1, B1 gamete, you have an A2, B1 gamete. 1-to-1 ratio. Thank you for asking. So we just talked about that second question shown here on this slide, how sister chromatid separation occurs during meiosis. First meiosis division, the 2 versions of the chromosomes you got from one parent, the paternal version or the maternal version, stick together. They stay together at the first division, then they separate at the second division. Question in the back.
I was going to ask, in the video, you got a 1-to-1 to 1-to-1 ratio, but in our example, because B1 and B1 are homozygous, would it be 25, 25, 50?
All right. So 2 points that weren't in the video that are going to be really important for finishing the next exercise, which I asked you to discuss or think about at the end of last class. This point's really critical. Spermatogenesis versus oogenesis. There's an asymmetry here that goes on when meiosis occurs. And I want you to know now, because I will not remember to tell you this again, probably. So write this down, let it stick in your mind. When I draw meiosis as looking like this, which sex is that? Is that males or females? That's males. And this is my default when I think about meiosis. I think about 1 cell division, 2nd cell division. And what did I do here that made some of you say this is male meiosis? I'm drawing 4 cells that are haploid, and how did I draw them relative to each other? Same size. Right? So when I think meiosis, I think 1 cell divides in half, and then those cells divide in half, and you get 4 equally sized gametes. That's male meiosis, where each of those things, if you're a mammal, gets a flagellum, and then goes out in search of an egg. Or dies waiting for the chance. What happens in female meiosis? We still start out with one, so here I'm going to draw in the numbers of chromosomes in each of those cells. You start with a tetraploid cell post-synthesis, post-S phase. You've got a cell that's got 4 copies of every chromosome, first division, second division. Every one of the sperm are haploid. What happens in the oocyte? 3 of the cells, I don't know if you could consider it dying, sure. Right, so what happens after the first cell division? Have I drawn this accurately? One might be a little bit bigger, but each of these first daughter cells after the first division are diploid. So up until this point, spermatogenesis and oogenesis are the same. Now what happens? One of those cells divides asymmetrically. So you have the oocyte, that's the primary oocyte, that's the thing that's actually going to be the functional egg. It's haploid. It's got 1 copy of every chromosome. What's this little nubbin over here? The one on the right, what's it called? It's called a polar body. Why does this look asymmetric? It's because that polar body has given all of its goodies in its cytoplasm to the oocyte. The oocyte is being loaded with messenger RNAs and proteins and other things that are essential for that oocyte to be a viable oocyte. That polar body that results after the second division is called a secondary polar body. And here's the primary polar body. How many copies of DNA, this is the most important point, how many copies of each chromosome are in the primary polar body? If the oocyte is haploid, and if the second polar body is haploid, where are all the other chromosomes? They're in the primary polar body. It has 2 copies of every chromosome.
And that's all from the top, this cell?
Right. So we start with the tetraploid presumptive oocyte, myocyte, divides in half, so you get segregation as normal, 2 copies of each chromosome in one cell that will divide again and produce an oocyte and the secondary polar body. The other polar body, the primary polar body, is diploid at that point. So after meiosis, or through the process of meiosis, you get the oocyte haploid. Secondary polar body will be haploid. And there's a point at which you can grab the primary polar body and it's diploid. Like I drew it up here. It's got 2 copies of every chromosome.
Oh, so wait.
Yep.
All it did was that cell was lowered?
Right. So that gets us to this question that I posed at the end of last class. I'll let you discuss this with neighbors in a minute, briefly. I'm going to give you a little bit more perspective on the question. And I talked to this with a couple of you in my office hours at the end of last class. So what was the point of this question? What's the concern?
How do you get the DNA extracted without destroying the quality?
Okay, so if you didn't hear that, I heard that. It's okay. I was actually surprised I remembered to turn down my phone before class today. No one ever calls me anyway, but just in case. Start calling me. Right, if you want to know something about, is this oocyte that was produced by a female, does it have an extra copy of a chromosome, for example, which you might not want to use if you're doing in vitro fertilization, because you could cause aneuploidy. You got 3 copies of which chromosome, you get Down syndrome? 21, it's one of our smallest chromosomes. right? So for example, when people are doing in vitro fertilization, they would love to know, here's this precious oocyte. Is it one we should use to fertilize? Or does it have a genetic predisposition to some disease? The concern then is, how do you do genetic analysis of that one cell? If you open it up to take its DNA out and study it, and you discover that that was the perfect oocyte, then what? You destroyed the oocyte by opening it up and taking the DNA out. So how do you do genetic testing for in vitro fertilization? That's the question. How do you genetically test an oocyte for quality and not destroy it in doing so? So that led to this. So please discuss these questions. What are the plotted on these axes with your neighbor? Again, 2 minutes. We'll come back together and we'll discuss what this graph means. This is data from an actual published study trying to accomplish the question that we just posed. How do you genetically test an oocyte? So go ahead and we'll come back together in 1.5. Let's pull it back together for a second. So again, if you're stuck or asking a question or having a good discussion, what point are you at right now? What are you talking about? What are the concerns? Things need to be clarified?
Well, if you take DNA from the polar bodies, you can still, it's supposed to be the identical DNA to that's in the oocyte. So you can use that rather than damaging the oocyte, which is what.
Right. So we have the oocyte. Where's the oocyte first? Let me number these. Panel 1, panel 2, panel 3, panel 4, panel 5. Top to bottom. What's on the x-axis? Right. Human chromosomes, 1 through 22, and then the X or the Y. Okay. What does 0, 1, 2, 3, 4 mean on the y-axis? Number of copies of each part of each chromosome. So this is essentially DNA amount. AMT. DNA amount. Number of copies of each part of each chromosome. Number of copies on this axis, chromosome going this direction. So the question is, this is a real study. These are data from a scientific publication where they actually took an oocyte and its polar bodies and genotyped. They took the DNA out of the oocyte too. Yes, they were just trying to prove the point that this approach could work. So they didn't care that they destroyed the oocyte in this case. This is just trying to prove to other people that you could do it this way. So what do you expect the number of copies of each chromosome in the first polar body to be? Here at the top, first polar body. Should be, as we just discussed, that first polar body should be diploid. So it should have, as their black line shows up here, it should have 2 copies of every chromosome no matter what. Was that true? What does that mean? It's aneuploidy. What does it mean when you've got, what's shown right here? This cell is trisomic. It's got 3 copies of, which chromosome is that? 16. Okay, well, thank goodness, because that's a polar body. It's not going to be used for fertilization anyway. So that's okay. Wait, is it okay? What's the question that you would have? If the first polar body has 3 copies of a chromosome, we started with 4 total right after synthesis. Now we're doing like the magician's shell game. Where is the 4th chromosome? What are the possibilities? It's either in the second polar body or it's in the oocyte. What do we want? We want it to be in the oocyte. Where was it? Yep, so there's 3 copies, I'm circling this in red. There are 3 copies of chromosome 16 in the first polar body, and there's 1 in the second polar body. So what's the genotype of the oocyte? The 3 copies of the chromosome in one polar body, 1 copy in the second polar body. What does the oocyte have? Nothing. And that's what this paper was proving, is that when they actually, that's the confirmed, down here, panel 4. Female pronucleus confirmed. That means the DNA that was taken out of the oocyte. they proved by sequencing that DNA that there was actually no version of that chromosome in that oocyte. Big shock. Arithmetic works. You start with 4 copies of a chromosome. You take the primary and the secondary pronucleus. You look at how much DNA they've got. And then you just do subtraction. 4 minus what was in the pronucleus, or 4 minus the primary polar body minus the secondary polar body results in what the DNA content of the oocyte is. You don't have to damage the oocyte to figure out its genotype. You take the DNA out of the 2 polar bodies, and then you subtract from 4. And a really interesting point was brought up over here while I was walking around. So is this an oocyte you would want to use? We decided on chromosome 16, the answer was no. It's lacking a chromosome entirely, which would be lethal. You wouldn't be able to get a viable fertilization out of that oocyte anyway. What else has happened to this oocyte? It's got, it's got 2 other issues over here, small chromosomes usually. So it's got an extra copy of chromosome 19, and it's also missing chromosome 22. So this oocyte has multiple genetic abnormalities. The scary thing is this is pretty common. This is a oocyte removed from a human female. It's not like they'd sequenced 1,000 oocytes and found one that was just really abnormal. This was just pick a random oocyte, look at its genetic quality. So chromosomal abnormalities and non-disjunction of chromosomes during meiosis happens a lot. And this is one way that geneticists can test for those.
What would be the scenario that causes that first bracket, for the first polar body, that first trisomy that we see? It's not a full chromosome.
Right.
It's all the case that one part of the chromosome got broken, never reassociated.
Right. So what this part, so the point is, here we've got all of chromosome 2 circled in blue, but only a part of that chromosome has been involved in a change in copy number between different cells, which suggests that the chromosome broke and part of the chromosome, part of one of the arms of the chromosome segregated into one, that is, was divided into one cell, and the other part of that chromosome went into a different cell, which also happens during cell division. You can have, unfortunately, chromosomes break, and then the 2 different bits of the chromosome get put into cells randomly, essentially. Follow up. Yep.
Where is that floating piece at? What is it? Does it just kind of like float around then, or is it?
So which panel? We've got a panel that's got too much DNA, the left side of chromosome 2, and we've got one that's got not enough.
So is it like, does it reattach to the second chromosome 2 in the or is it just kind of float around?
So what happens to that chromosome after meiosis, in other words? What happens to a piece of DNA that doesn't have a centromere and 2 telomeres? It gets degraded usually. So if you lose part of a chromosome arm, usually you've lost it for good, unless it happens to reattach itself to another chromosome somewhere else. Yeah. One more question? Yeah.
You said this happens a lot. What would be like the percentage, the number value?
Yeah, so how frequent is this a lot that I'm talking about? I actually don't know, but I will look that up for next class. This paper, let's see, when was this published? 2013. So it's only been 3 years since scientists started looking in earnest at how frequently these sorts of events might be in normal meioses. So it's not a lot of time to actually rack up that sort of number, but I'll look into it. Okay, so that's meiosis. To wrap up and to lead us into next class, I want to show you a few videos. We're going to motivate what we're going to talk about when we start talking about transmission genetics, how traits move from one generation into the next. So I want to introduce you to my favorite worm, C. elegans. So this will just be brief. Please keep the noise level a little bit low. Look, this is what C. elegans normally look like. They crawl around, we grow them on auger plates. This is not my video. They just move around like this. They crawl in a sinusoidal motion. They look kind of like snakes. They crawl around. That's normal movement for C. elegans. This, on the other hand, is abnormal. You may not be able to tell the details. This is called a roller mutant. They've got a genetic defect that instead of crawling from side to side like a snake, they sort of spin as they crawl. They're rollers. That's why we call them roller mutants. Hopefully nobody was videotaping me doing that. Right, and they don't crawl in a straight line. They tend to crawl in circles because they're spinning as they're crawling. They get stuck, basically. They just go around and around and around and around. We can also have worms that we put green fluorescent protein into. So these would be transgenic worms that scientists have artificially added a jellyfish gene that glows green when you shine a light on the worms. So you could, if you really wanted to, make a green fluorescent roller. And I'm thinking, as I proposed in the lecture slides, maybe you want to generate those sorts of worms for selling at raves or something. Green roller worms. So this is the idea for next class. A roller worm, last minute, I promise. The roller worm, if you've got one mutant copy, one mutation, these are diploid individuals. So you've got two copies of the gene that lets them crawl normally. You could have one version that's mutant, that's the minus, and one wild type, that's the plus. That doesn't make you a roller. For the worms to spin when they crawl, you have to be minus over minus. You have to be homozygous for the mutation. For GFP, all it takes is one copy of the GFP gene for you to glow. You don't have to have two copies of it. So one of your chromosomes can have this gene, and you're green. The question is, here are the genotypes of some worms you have. You have one strain that is homozygous for the roller mutation. You have a second that is heterozygous for GFP. That's the asterisk, the GFP. So think about these three questions for next time and come prepared to discuss, could you create that worm we just saw in the movie, the GFP green fluorescent roller, and watch one movie for next time in addition to the readings. Okay? Office hours now for an hour.